Our world abounds with symbiosis. In the flowers and the motley crew of animals which pollinate them, in the intricate mycelial meshwork, which connects 90% of all plants to soil, in the coral reefs, which house 2/3 of all marine life, even in the very cells of our own bodies and all of complex life, which evolved 2 billion years ago. These foundational relationships, these key stitches weave the foundations of our world, and we neglect them at our peril. Our peril in the ivory bones of bleached corals, the brown needles of decayed forests, abandoned by fungal symbionts, plants flowering to find no one buzzing to carry on their genes. All ongoing incidents foretell what is to become commonplace if we refuse to listen to the intimate relations of life, including our own. When the keystone falls, the arch crumbles. When the key stitches snap, we all become undone. My name is Benjamin Felser, and I'm an undergraduate biology major at Bowdoin College. Over the past year, I've been working on this project, Key Stitches, Symbiographies for a Distressed Earth, and writing about 4 key symbiotic relationships and a lot of different ecological ideas around them. I've been exploring how they evolved and how their stories can inform our responses to ongoing environmental crisis. Much of this damage results from Western culture leading us to perceive humans and other species as individual objects instead of as relational entities. Each of these symbiotic bonds I mentioned offer lessons on how to engage healing by changing the stories we tell about the world. My ideas are directly inspired by engaged artists and writers and scientists like Potawatomi Nation member Robin Wall Kimmerer, like Octavia Butler, Shayna Steingraber, Rachel Carson, and other engaged scientists and writers whose work is just as urgent now as ever. We'll explore these bonds through poems, which are a piece of the project that I've been creating. Starting with Twinkles at Dawn and the origins of complex life, onto Intentacled with algae and their charismatic corals, mycorrhizal openings with mycorrhizal symbiosis between plants and fungi, and blossoming with plants and animals and intimate pollination. I hope you'll indulge a little creative license and explorative personification, but also know all of these stories are informed by arduous literature review, and I don't pretend to know what it's like to be any of these organisms, but it's a useful exploration of how to open our minds, even from a scientific context. We'll start with eukaryogenesis, the origins of complex life, where a bacterium and another unicellular organism called an archaeon merged following the Earth's first great extinction event. Slowly in slime. It's strange how in primordial depths, one of the most earth-morphing events emerged slowly in slime. At the bottom of murky waters, amid twinkles at dawn. 2.4 billion years ago, bubbling gases emitted by biology. Toxicity bubbled into being as most life died, intoxicated by the novelty of photosynthetic emissions in an ocean that had never known oxygen, not at this intensity. They cycle back, falling into fuel for adaptive biofilm, bumptious layers of sedimented life, revolving, exchanging food and waste, outbreaths and inbreaths in orbit. Diffuse lights shimmer through murky depths, motes twinkling, subsumed in cycles through intermingling cells, as oxygen presses sensitive life deeper, opening space for adaptive possibilities of those capable of change. As tolerance becomes an asset, those below press up against the changing, intermingling metabolic pathways. Orbits condense as archaea consumes bacteria. Genetic legacies of histories apart merged into a singular lifeway. To wriggle up from muck into open oceans of possibilities. The first eukaryotic life. To become trees, beetles, fungus, jellyfish, elephant, humans, yeast, chimps, but for now, just another dust mote twinkling at dawn. We see here that it's only under the stress from the first great extinction event, the great oxygenation event, that eukaryotic or complex life emerged. Bacterial and archaeal metabolic paths became so intimate in response to this pressure that their cells literally merged and something new proliferated. The origin of species through symbiosis or symbiogenesis grounds the following relationships we'll explore. Championed by scientist and writer Lynn Margulies in the mid-late 20th century, but originating decades earlier, this overlooked theory implies that the relationships at the core of speciation may also spell their collapse when sufficiently disturbed and neglected. No one questions the disorder. No one questions the dangers of disorder between the mitochondria and the rest of the cell. But we still fail to recognize relationships in the contemporary conservation issues of our day. The story of corals and the symbiotic algae, which evolved with them, will help elucidate what it means to lose that connection. Within my bubbled home, I rest, remembering times when things were less certain. When we floundered, feared, and flew in search of a home we didn't yet know. Sometimes I remember how this intimacy was born from catastrophe. How warming waters stratified nutrients, how it sequestered below all we needed, starving, suffering in this stress. Vast possibilities upwelled and water stripped of sediments. Clear sunlight pouring through novel possibilities for corals and algae in need. We weren't digested, but ententacled in a coral embrace. They fed us nutrients as we offered sugars, bonds terraforming through the intimacy of time. Alongside millions of microbes, fish, eels, sharks, urchins, algae, fishermen, crabs, among, beside, inside, above of us. And all I need to do is live and breathe. Breathe and share the fruits of my life, spinning sun to sugar. You can't imagine the pain of knowing, of watching oxygen manipulate my controlled enclosure, invoke chemical wrath, the pain of knowing there is no other way for me to respond in this heat, my being is simply toxic. So the bubble constricts, coral compresses, and I've become hurtled into the unknown. As the home beneath me, all I've ever known becomes bleached. Skeleton, ghost. If only I could have known how to scale back, to slow, to stop poisoning the only home I've ever known. The home that without me is a ghost. If only I could have known. You know, you see behind eyes shuttered closed, behind those silk thin veils, but you distance in time as you poison in space. Bleached earth abounds in your present. If only I knew how to tell you what is coming if you don't listen. You don't listen. You don't stop pumping heat outward, stop dumping your waste, so you choose to forget within your bubbled home. Our negligence reflects an inability to understand how symbiosis exists beyond this coral bond. To be clear, our refers to myself and others in the global north, but especially militaries and petrochemical companies responsible for 92% of global emissions driving ocean warming and coral bleaching. Their collapse disproportionately impacts people in the global north, the 500 million people directly dependent on reefs for food, income, tourism, meaning, and storm protection. Inevitably, it'll ripple to us all. Maybe by rooting, our roots can branch through mycelial networks to tap into our inherent interconnection with non-human worlds. And so we turn to plants and their mycorrhizal or root symbionts. resting in my carbon fortress of lignin and cellulose. I release treasures into open root tips, burrowing within their cavities, leaning into the fibers which interbraid me. I sneak through crevices in search of captive nitrogen, phosphorus. Recalcitrant resources I transform into sumptuous offerings. Your roots beckon. Come here. I'll guide you through to the core of nourishment. We exchange ancient signals of intimacy. Yes, I am who you think I am. Sacred sanctum where I unburden myself of offerings. I have something for you, I call. I brought it just for you, if you'd like. In response, a flood of carbon, stuff of life, rushing like endorphins to overflow my hyphae into vast networks beyond crevices, around corners. Overflowing abundance. It sprouts, exudes from my threads, feeding the bumptious fray of bacteria who offer me micronutrient nourishment, who encompass me, guiding me to root, defending my networked being. They clamber across my form to find new lives, new homes, El Camino Real for microbial migrants, searching. And here we carry evolutionary legacies of exchange. Carbon careens about empty skies, exhaled by open mouths, inhaled into gaping leaf tips, spun into crystalline sugars to be shunted down towards open roots, into open threads, branching into earth, into me. But what if you didn't need me? What if you disappeared? What if suffocating abundance plugged all sites of intimacy, as fertilized hyperfecundity obviated the need for mutual aid? And I'll starve as you choke on the promises of a green revolution. But how much you lose in pursuit of increased yield. You know I've always protected you from pestilence, how I sucked water when little was near, how I held the soil sticking around your vulnerable form as you grew. What waste to see so much slough away alongside my threadbare corpse in the rains. How you suffer in my absence. Agricultural industrialization, evoking realizations of how much we need each other. My spores waiting, resting patiently to again branch within your carbon walls. Mycorrhizal networks weave the foundation of terrestrial ecosystems and have for over 500 million years. But twin threats of excess nutrient runoff from excess fertilizer use or eutrophication and habitat destruction itself threatened this key stitch. Fertilizer use neglects the ancient legacies of dialogue between fungi and plants. And though necessary, negate the reasoning now that there are other ways. Mycorrhizae form vast subterranean networks known to increase water and nutrient use efficiency, protect against pathogens, and they reduce erosion rates by up to 400%. With fertilizer use increasing by 500% since 1950, and both logging and fires deforesting 2.5 million acres a year, neglecting mycorrhizal symbiosis is disastrous. But for now, turning to above ground networks, we'll see how plants and pollinators may show us what can flower in our current moment of catastrophic change. Blossoming, expanding into the fullest form of myself, flourishing incandescent, an aromatic exposition of all I've come to be. Is this what bees smell? My forms of bees, moths, bats, fluttering over parched skies to islands of enflowered vitality, buzzing, clambering, flapping vehicles for genes, propagating pollen granules grounded in my moth fuzz, bee fluff, bat fur. Sometimes I remember my vibrance all those eons ago. 200 million years ago, the first flower bloomed in the damp, dark depths of the Triassic jungle. Flowers no larger than a single period, enjungled by giants who made home for me in their shadows, homes where others had been upturned and I could root, clinging to specks of life, brief openings in the canopy that shielded me, muffled me. As parts of me learned flowering, insect pieces learned that floral nectars, fecund fruits could be just as nourishing as leaves or decomposing flesh. Where do I grow when the world opens before me? I flowered in the shadows of upturned earth, but now all is unearthed as magma and meteorite birth flooding fires, a year of winter under ashen skies, under hundreds of meters of calcified ash, still latent under millennia of soiled sedimentations. Just 67 million years ago, ending the reigns of dinosaurs, 80% of all earthly life gone. Reigns of non-flowering plants older than terrestrial ecosystems vanished. This closest great extinction, this closest boundary between what was and what's become, what we've become. Sometimes I wonder how many flowers blossomed in the eaves of those ashes. Sometimes I'm nostalgic for those who waved to and fro above me, who shadowed the expansive blueness of sky. Shadows of shadows still linger in my memory, and how my leaves grow broad, and how they leap from direct sun, afraid of its excess, and how things have changed. Here I sway, buzz, flutter, flower, float, crawl into fluttering, buzzing, flapping emptiness, where there was a beacon at this time last year, a flower. But rains came early this year, too soon for heat to signal time to bloom. And so I flutter in languid loneliness of what remains, descending lifeless into earth. Hopefully it's different next year. It's moments like these when I remember those damp, dark depths and those first flowers under dense canopies, when I first graced myself in pollinating pleasure, blossoming. As over half of non-ice Earth is converted to homogeneous agriculture, to homogeneous agricultural fields, and pesticide toxicity increases by 300% in countries like the US, changing environmental cues may be the least of pollinator concerns. They terraformed the world after a meteorite opened our current age, and we stand at another turning point. But we turn too fast for biological evolution to respond, at least without gargantuan losses. Unlike corals and plants, we can choose whether or not to expel our symbiotic cohabitants. We can choose how to tend to these key stitches, how we can regenerate through change, through symbiogenesis, through taking that to heart. Human presence is not antithetical to environmental health. These relationships emerge through the reality that precarity is better met together, and we have the capacity for change. These key stitches tell stories of a symbiotic past, and our futures will depend on how closely we listen. Thank you all for listening, and thank you to GRC for putting this together. I'll be releasing a full suite of my writings, which also include more traditional science writing pieces, if that's more your style. It'll come out on my website on May 28th. So please email me if you'd like an update whenever that's ready. Again, thank you all for your time, and I hope to talk to you in some of our more unstructured conversational dialogue. I hope you all have lovely days.